obvious differences between the weedy rice germination patterns. When temperatures are between 25 and 35, there doesn't seem to be a big difference between water potentials between 0 and negative 0.6. Excuse me. At temperatures below 25, water potential seems to play a greater effect. It seems to play a greater role on seed germination, and the more negative the water potential, the slower the germination rate. Okay, so now I'll move to the next chapter. I just want to remind you that what we just went through was the data that was collected for here. And now I'm going to sort of move through the data that's collected to create this half of the curve. So for this chapter, my objective was to determine emergence patterns of California weedy rice over time using soil and flooding depths. So besides the modeling aspect, this experiment was meant to challenge the cultural assumption that a deep flood will reduce the growth of weedy rice. So to conduct this experiment and kind of test that hypothesis, we again used our M206 control and several weedy types. We used a range of flooding depths and burial depths. We created a factorial split plot in RCV in a randomized complete block design, and we had 4 replicates per experimental run. I would also like to give a very nice shout out, even though he's not here, to my current lab mate, Dennis, who's currently doing a PhD. He was very, very important in helping get this study off the ground and running. So with that, oh, and another important, we collected cumulative soil water emergence daily, as well as dry weight. We also recorded temperature over the entire experiment to help translate into those thermal time models. So for the results of this experiment, we're seeing that burial depth had the greatest effect on weedy rice emergence. Nothing buried at or below 5 centimeters emerged, so it was not included in our final analysis. There were significant interactions between burial and flooding depth. So what we have here is an increasing flooding depth in centimeters and our average emergence percentage on the y-axis. This dark colored bar represents a shallow burial depth, and this gray bar represents a bit deeper in the soil, 2.5 centimeters. So obviously there are significant differences between these 2 bars, the average emergence from 1.3 and 2.5 centimeter burial depths. I did not see as dramatic a decrease in emergence with increasing flooding depth compared with increasing burial depth, okay? So now here on the right, we're trying to determine if there are differences between emergence from the soil and emergence from the flood water surface, so the soil and then the flood, right? So what we have here is, again, the increasing flooding depth on the x-axis and the average emergence percentage on the y-axis. Here, the dark bar is the soil surface emergence, and the gray bar is the water surface emergence. So this graphic is showing you that if the seeds are able to emerge from the soil, they are also going to emerge through the floodwaters. Flooding did not stop growth once the seeds emerged from the soil. So our conclusions are that keep in mind that the conclusions from this study are that the best emergence occurs when seeds are buried at shallow depths and there's also shallow flooding. Weedy rice will likely not emerge from depths deeper than 5 centimeters. And contrary to popular belief, flooding depth alone did not reduce weedy rice emergence. And if weedy rice does find its way into a grower's field, it's likely to produce more dry weight over a short period of time, which means that weedy rice can become more competitive more quickly. Okay, so that was chapter 2. Now I'm going to go into chapter 3 where I'll talk a bit more about the modeling aspect that I've derived from these last 2 chapters. So to kind of set up the objective for chapter 3, UC Davis currently uses degree day models on different University of California websites for assisting growers in various ways. So here I'm showing a photo of the Agronomy Research and Information Center for Rice, where growers can estimate the timing of growth stages of cultivated rice varieties based on the cultivar, which you see here, the planting date, active station means they're collecting weather data and temperature data. And then they also have the option of putting in where their field is and the acres that they have. So ideally, the models that I create will be used on a similar website and will be used for assisting growers in determining the timing of weedy rice emergence so they can time their controlled applications appropriately. So the model that I would like to create, instead of planting date and active station, they would put in something like weedy rice type, flooding depth, flooding date. They might know the seeds in the soil seed bank, etc. So in order to create this, my objective for this chapter is to compile cumulative germination and emergence data into a predictive thermal time model and validate the model output with field level data. So please keep in mind, I have not created the models yet. However, I have collected all of the field data. And before I show you how we collected the field data, I would just sort of like to show you this conceptual methodology for how the model is going to be created. So here what we have are the germination and emergence studies. So the brown represent the inputs and the outputs from the germination study, and the green represent the inputs and the outputs from the emergence study. These are all labeled with some type of symbol. Each of these symbols represents a mathematical placeholder that will be determined once I have completed all the research from these two experiments. So once we have created the mathematical model from our theoretical data, we have to test that against actual field level data. So if we only use lab data or controlled study data, we actually don't have a true picture of what's happening in the field, hence the necessity to collect this field data. So what I'm going to show you in the next few slides are the results from the 2019 field season. So to set up our fields, we simulated a seed bank by seeding the field with weedy types 1, 2, 3, and 5, which were planted in rows, which you kind of see here, in the fall of 2018 and then in 2019 across the entire field. Starting in 2019 and then again in 2020, I created the sampling points, which is what these red arrows are pointing at. These are actually buckets, which had the bottoms cut off. So they act as a collar. So debris can't flow into the sampling point, but water can seep in through the bottom. So the flooding level is steady, but my sampling point is a specific point in the field, so I know what data I've collected. Okay, and because our collection points were kind of small, each bucket was planted again at random depths with 30 seeds to ensure we collected enough data. These seeds were not pre-germinated and they didn't have the dormancy broken. We also used these data loggers that you see here. You can't see the soil logger because it's buried in the soil, but this is kind of what they look like. Each row had its own soil and temperature logger, and they measured temperature every hour each day for 28 days after the flooding was applied to the field. And just so you know, I would reach down into this bucket, and this is what I was looking for. Some tiny little seed, and I would have to try so hard to find them. And sometimes it was cloudy, so it's a very exciting field experiment. So now I want to kind of show you some of the results. Here on the x-axis, again, we have days, but because I recorded the temperature, this will be converted into thermal time. And then on the y-axis, again, I have emergence from the soil up to 100%. These different bars represent the different weedy types. I did not use M206 in this experiment. So our type 3 is the purple bar, type 5 is the orange, type 2 is the blue, and type 1 is the yellow. So from this experiment, what we saw is that most weedy rice emerged from the top 2 centimeters in the soil, which is what we saw in our emergence experiment. And very few seeds, if at all, emerge from depths below 2 centimeters. Type 3, which is suspected to be the type that has existed in California for the longest amount of time, has had the highest emergence, almost 90%. Okay. So before I wrap up my talk, I do want to present some contradictory evidence to my own findings because I think this is a very important thing for scientists to acknowledge, that we're not always 100% right. So just because we're seeing seeds emerge from the upper 2 centimeters doesn't mean there aren't other seeds emerging from different depths under different flooding scenarios in the field. So what I'm showing you here on the right is a type 3 seed that was not within my sampling point. I just saw it in the field. It had already emerged through the water 9 days after flooding, which was much faster and earlier than the seeds that I had within my sampling points. This is likely due to the fact that we had an abnormally rainy spring and the rains went very, very late. In California, we have a Mediterranean climate, and so it typically stops raining in early April, late March, and then the rice preparation begins. In 2019, it rained very, very late. And so I can't say that the flooding we applied was why this seedling emerged. It could have had the right temperature and soil moisture from the rain, and then when we applied the flood, it was already ready to come through the soil. So I throw this in here because it is an example of how error is created when trying to apply theoretical models to actual field settings. You will never capture all of the scenarios in the field and will not be able to adequately predict them with a mathematical model. However, what we are attempting to do here is to capture as many scenarios as possible with as many replications across seasons to ensure that our final model is reliable enough for growers to use. And with that, I would really like to acknowledge the UC Davis Weed Science students and staff who have helped me a lot with this experiment, as well as the farm advisors or extension agents that I work with, and the Rice Experiment Station and staff. I also would really like to thank my funders, the California Rice Research Board, the California Cooperative Rice Research Foundation, the D. Marlin Brandon Rice Research Fellowship, the Rice Experiment Station, and last but not least, my fabulous...
The first speaker, so we have few questions waiting for her. Liberty, there is one person asking, Syed Tausif, you want to know more about stale seedbed.
Okay.
Could you tell us or elaborate more about stale seedbed?
Sure. Is it possible for me to share my screen to help answer the question better?
Yeah, sure.
Okay. Let me, excuse me, just a moment. Okay, so I think there were two questions. One person asked about organic production, like weed control in organic production, and then someone asked about stale seedbed. And actually in organic production, you can use a stale seedbed for your weed control. So I wanted to reiterate this particular slide. Can you see this one? So the stale seedbed, I think a key part that I left out of this particular slide is that many seeds, regardless of whether they are on land or in the water like rice, they require water or imbibition in order to germinate. So what we're seeing in this slide is these red triangles represent the weed seeds, and in my case, weedy rice. But keep in mind, this scenario, the stale seedbed can be applied to vegetables, row crops. It can probably be applied to orchards and vineyards, but I don't know as much about perennial crops. So the idea here is that when you apply your irrigation to the field, there is enough water that goes down into the soil that causes all of these little seeds to germinate. And so then after they germinate, they go towards the sunlight, they go towards the soil surface, and the grower just guesses. They say, well, there's a lot of weeds in my field. I guess it's time to spray them with herbicides, which is what you would do in a conventional operation, or in an organic operation, they can go through and do shallow tillage to try to cut the roots or cut the cotyledon off at the soil surface. So after irrigation causes these seeds to germinate, they come up, and then depending on if you are organic or conventional, you can then control the weeds in whatever manner that you want here. After that, you can then flood again if you're in rice, and plant your crop. Now, a really important thing about the stale seedbed for any scenario is that if you are constrained by time, so in California, we have to plant our crop in March or in April or May, because at the end of the rice growing season, the rains will come again, and the rains cause all of the rice to fall over if the rice is not already harvested, and then it's very difficult to harvest. And so what happens sometimes is in the spring, if it rains too late into the season, you don't have enough time to implement this whole stale seedbed methodology, because you have to prepare the field, you have to apply water, wait for the seeds, then you have to totally dry the field again, do your control strategy, and then flood the field again. And so this particular strategy is very dependent on weather. There's some really, I will post in the chat box, there's some really good references that I found for the stale seedbed. So if anybody's interested more in that, I'll post those. Stop sharing there.
Thank you, Liberty. The next question comes from Pintu Telem. He is asking, what kinds of instrument or protocols are used for germination and quantitative analysis of water measurement?
So I can answer these questions. Sarah has also done a really good germination study. So Sarah, if there's something here that I missed, which I'm sure that I will, please jump in. But to measure our water potential, we use a polyethylene glycol solution that we mix to whatever concentration we want. So negative 0.4 has a specific amount of polyethylene glycol that you mix with water. This is actually documented and referenced by Hardigree and Emmerich, which I'll also put that reference in the chat box. So you mix your solution, and then essentially what we do is we mix up the solution. We use a water potential meter to make sure that our solution is at the right concentration. And then we soak filter papers in the solution. And then the filter paper goes into a Petri dish, and then the seeds are grown inside of the Petri dish, like I showed in one of those slides. So that's essentially how we create the different solutions. And then we were growing those inside of a chamber. So for the field aspect, I'm actually not testing those different water potentials. One of the field researchers that I'm working with is doing that particular portion of like the field validation of that study. So for my research, I'm only testing the full field saturation level. Yeah, is that good, Sarah? Did I miss anything?
Another viewer has asked, how can we control weeds in rice fields without using weedicides or chemical fertilizers in organic farming?
So I know that in California, and I'm sure it's very different in other parts of the world, but I've talked to several organic growers that have told me that they actually switch their fields. So one year they'll grow rice in field A, and then in the following year, they won't grow rice in that field. They'll grow it in this field. And what they do during that year when there's no rice here, they go through and they do a lot of cultivation, or maybe they'll spray an organic herbicide. And when they are growing rice in this particular field, they use a 30 centimeter flood. That's a very common practice. And the idea is that the very deep flood, this 30 centimeter flood, will reduce the weeds. But I actually have some, I'm a bit skeptical that these deep floods will kill weedy rice because it's so similar to cultivated rice. Yeah.
Okay, good. So at the end, I would like you to give few inspirational words for the young researchers out there who are listening your talk.
Okay.
The young what are your words of advice for the young researchers?
Sure.
So yeah, like you?
Yeah, like me. Yeah. So I, the academic system in the United States is very different from other countries. But I would have to say, I think probably from a mental perspective, my biggest piece of advice is don't be afraid of rejection. So if you find a person or an idea that you really want to do research, like you want to do research with a person, or you have this idea and you really want to explore that idea, you know, go to that person and ask them if you can study with them, because the worst thing that they could say is no, and then you are right back in the place where you started. And if you don't ask that person, or if you don't explore that idea, you will never know if that was an opportunity for you, or if that question is answerable, or you know, anything like that. During my master's degree, I was rejected a lot by a lot of people, and it took me a really long time to find someone who I guess kind of believed in me and supported me. And now I'm doing a PhD alongside Sarah, who's a great researcher, and also Kasim Al-Khatib, our professor, is incredibly supportive. And I would have never done a PhD with him had I not been rejected by many people, and then finally accepted, you know, asking and asking and asking and trying and trying. So that's my advice.